Hello and welcome to this video, Death by Cucumber, the psychology, computational architecture and molecular biology behind cucumber phobia in cats. First of all, I don't even know if cucumber phobia is a real word, I'm just coining it right now. So probably like many of you that have seen the internet phenomenon that's been spreading around, cats have this interesting response to the placement of a cucumber behind them when they're eating, and you know, some people have just found this funny, and others have asked why is this happening? Why do cats behave like this? First note, not all cats behave like this. Some just are inquisitive and curious about the object, and even others just ignore it. But here's a possible explanation that goes probably a bit too much in depth as to why cats respond this way. But these kind of things, they get stuck in my mind, and I'm up at 2 o'clock in the morning thinking about it, and I just have to find out why animals behave this way. So here's the video, and I hope you find it interesting as an explanation. So the first thing we need to discuss is evolutionary psychology. What is evolutionary psychology? Now, going a step backwards towards evolution, perhaps some of you may know that traits such as fur texture, fur patterning, colour, the shape of an animal's body are adapted to the environment. What this means is particular systems or organs have increased the reproductive success of an animal in the past by enabling them to leave more copies of their genes that code for that trait in the next generation. Which is to say that in, for example, the giraffe having a long neck, and one individual had a mutation that led to having a long neck, and this individual was able to reach taller trees and eat food, and so it outcompeted the other giraffes in terms of reproductive success in its population, and so it left more copies of the long neck gene, and we get these amazing badasses. But obviously before it had the long neck, it had all the other parts of its body, and the long neck is just an extra trait. It is a tinkering on the current design of the animal. Each piece, each adaptation that the giraffe has, these antlers which it uses to compete with other males during the mating season, large ears to release heat, they didn't all arrive in terms of the animal having the trait, having a mutation, at the same time. They're additions that happen throughout the evolutionary history of this organism. Now, if your mind is a biological organ, your brain, it's the function of your brain working, and it's an organ just like the rest of your body, then mental abilities should be traits that get passed on to the next generation in all organisms, including humans, because we are animals too. And more importantly, those traits shouldn't be there all at once. Your mind is a collection, it is a zoo of all these different traits cohabiting the neural structure of your brain. And notable examples in humans are facial recognition. We're going to discuss that at the end of this video. Our ability to language. We can acquire language unlike other species. We seem well adapted to it. Spatial mapping. This is something we share with rodents and many other mammals. And a capacity for social learning and acquiring culture. Now culture, yes, culture shapes our minds, but we have a capacity to be enculturated, to learn culture. And so our mind is something like a Swiss Army knife with loads of different tools on it. And that brings us, with evolutionary psychology elucidated, we can now discuss why the cucumber phobia expressed by cats. Here's just a brief showcase of the cucumber stress response. they turn around and they're not expecting anything to be behind them, because when they started to eat there was nothing behind them, and then suddenly there's a cucumber behind them.
Okay.
So as a scientist, I couldn't help but try this out on my own cats. So Pepper is the one on the right or below, and Maisie's the one on the left with the white markings on her face. And I'll place this cucumber here. Now, retrospect, having the plastic still on it probably might have affected their perception, as well as the orientation of the cucumber. So as you can see, Pepper is the first to notice the cucumber, and she does give a slight shifted startle response there. Then she simply investigates the cucumber a bit more, decides it's no longer a threat after collecting the information, and then leaves, which catches Maisie's attention. She looks in the opposite direction, and then she has a little startle response, which is a bit delayed, so I don't know if she was daydreaming or processing the information a bit slowly there. Thank you for taking part in my experiment. Good girl. Well, given our idea that the minds and brains of all organisms are like Swiss Army knife tools that are adapted to specific niches, solving specific problems of the environment, we can go ahead and suggest that maybe the reason why cats are afraid of cucumbers is that they experienced predation by snakes. Snakes resemble cucumbers, and cats that had some cat in evolutionary history that had a mutation that led to a computation in its brain that detects predators like snakes, was able to leave more copies of its genes in the next generation than all cats today should have what I call a predator detection module for all sensory stimuli that look like a snake. So this is an evolved psychological adaptation. Why would all cats have this? Well, we can look at the evolutionary history of cats and their the ancient environments that they would have interacted with. Cats are part of a larger phyletic order called the Carnivora, and Carnivora begin with the Myocoids 33 million years ago. And the modern domestic cat is has a common ancestor which occurs much later in evolutionary history, 10,000 years ago, and this ancestor is the Near Eastern African wildcat, and we have evidence for the first domestication of the Near Eastern or African wildcat in the pre-pottery Neolithic period of 10,000 years ago in Cyprus. And so this is the ancestor to all domestic cats, which their biological name is Felis domesticus. And speaking of Cyprus, one of the reasons why cats were introduced to the island from the Near East was actually that they had an uncanny ability to defend themselves from snakes, and this is why they were brought to the island to help decrease the snake population. So that kind of goes forward to our idea that, you know, maybe cats have this innate recognition for snakes, for the threat that snakes pose, and for activating a flight or fight response to either flee from the snake or disable it as a threat in its environment. So having an evolutionary history in which cats have been exposed to predators like snakes, from being possibly going back as far as the myocoids, which look something like pine martens, or even something more recently, such as the Near Eastern African cat. Being exposed to snake-like objects generates a statistical regularity, that there is a snake-like feature in the environment, and this means that any mutation that enables the cat's brain to instantly recognize the snake and do so automatically and without interference would increase the reproductive success of that cat and pass on this innate recognition, our PDM, the predator detection module, because of this evolutionary environment of adaptation, or the EEA. So the PDM's function is to defend the cat from this EEA, in this case, the snakes. So why is the PDM activated by cucumbers? Well, obviously a cucumber might resemble a snake in some ways, but in order to understand why the PDM interprets the cucumber as a snake, we need to think about brain biology and the computational architecture of the cat's brain, and that's what we're going to do now. So first thing you need to know about the nervous system, the brain, is that it's comprised of these specialized cells called neurons. Now here we have a schema of the peripheral nervous system, so this is in your spinal cord, and what you have here is some tissue, the dermis. This is your skin, and it has sensory receptors in it so that you can sense touch. So if you were to touch the wall, for example, sensory neurons, these specialized cells here, the mechanic abrasion of your skin against the wall would activate receptors in what are called the dendrites of this cell, these little processes here, and send electrical potentials down this shaft called the axon, which is a lot like a wire, and propagates the signal towards these terminals. At these terminals, a chemical signal is then released in response to the electrical signal and crosses small gaps over to the dendrites of the next cell, and the electrical potentials, called excitatory postsynaptic, meaning post-gap, the next neuron, travel down the dendrites of the next neuron and cause another potential in this interneuron here, back down the axon to the axon terminals to the next neuron, and this is a motor neuron. The electrical signal goes down the axon and down these axon terminals and onto a muscle fiber that causes contraction. So a better example in this case would be that you've touched something hot and the signal goes in a reflex arc. So there's the sense of the heat, the interneuron to the motor neuron, contraction muscle, and you pull your arm out instantly away from the hot object. You do it without thinking, but this same kind of network structure is what you can find without the motor neurons when you're doing declarative thinking, cognition, and recognition. With sensory neurons like that of in the eye, the retina, in the case of the cat. Looking at this more deeply so we can get an idea of how neurons can come together to compute things, to enable you to think. An important thing to realize is that neurons are biological. they are natural, and as such they're not like computers. They are a good analogy is that they are bags of chicken soup, of salty chicken soup, with more soup around them, the tissue fluid around them. And in the tissue fluid are salts. I've got these balls representing sodium ions, sodium as in salt. And inside the neuron are also some more balls which you can't see because they're inside the cell, called potassium. Now sodium has a positive electrical charge, and what happens is when the signal comes from a presynaptic neuron here and sends a chemical messenger that binds to these dendrites, it causes a mini electrical signal that comes down these dendrites. And of course that electrical signal has a positive charge. Now if enough of these signals come down these dendrites and increase the internal voltage, millivoltage of this axon hillock here, at this point here, from minus 70 millivolts to a threshold of minus 55, so it increases it enough, little sodium channels inside this axon will open. They open at minus 55 millivolts and enable these blue dots, the sodium ions, positive charge, to enter the cell. And what this does is it increases the inside voltage to plus 40 millivolts, or positive charge, which is what we've got here. These positives meaning that the sodium has flowed in and it's depolarized the inside of the cell, which means it's just flipped its charge from being negative inside the cell to being positive. And this flipping travels down the axon, and that's what the electrical signal is. But you have to have enough inputs to increase the the internal voltage from minus 70 to minus 55. And what that means is that this neuron will only fire if it has enough stimulus from neurons around it. Now this is really important for building a biological computer, and this was realised by McCulloch and Pitts and is shown in this diagram here. So this is more of a digital computer version, a simulation of a neuron, if you will. So this circle here is the neuron that we're interested in, and these smaller circles with x1, x2 and x3 and x4 are other neurons. In this case we could call them the sensory neurons from back when we were talking about touch. Yeah, this is one sensory neuron. Here we've got 4 sensory neurons, and we've got here this big circle can be this interneuron. And what we can say is that this interneuron here is now only going to fire when it gets signals from several sensory neurons. Now these sensory neurons are sending their axons to this neuron, and these axons have synapses, and the strength of the synapse determines how much this sensory neuron is going to stimulate this interneuron. We call this the weight, the strength of the connection. Now the threshold for this interneuron here is 4. Now we had it at minus 55 here because that's the biological truism of it. On average, that is the threshold of your average neuron in the body, but this is a computer simulation, so we can just say 4 arbitrarily. Now neuron 1 has a weight of 2, which means when it activates and it goes from being 0, an off state, to being 1 in binary, activating, switching on like a switch, we times this by 2, so the strength of the signal is going to be 2. This alone is not enough to trigger this neuron to fire because its threshold is 4. This neuron is only a 1, so that's not going to do it. In fact, none of these neurons alone are going to do it. If neurons 2 and 4 fire, 1 and 1 plus together is 2, it's not going to fire this neuron. But neurons 1 and 3 do, which means that this neuron, this interneuron, is very good at recognizing sensory stimuli that have features that are activated, that activate sensory neurons 1 and 3. That's basically how neurons compute. That is, sensations can be categorized by higher level neurons. and this here is basically the threshold function, which is to say that if these inputs sum, when you add them up, this is the sum function, when you add them up, if they are higher than the threshold of 4, if they are high enough, if they are a positive number, they switch to 1. This neuron then activates and fires, and sends a signal down its axon. This means that neurons can compute with what's called Boolean logic. Don't worry about that term, that just means that they basically can be used to make a computer. In your computer, you have these things called logic gates, which do all the things that you want the computer to do in terms of its hardware. Okay, and the software of a computer, your apps on your phone, for example, are the information stored in these zeros and ones in a binary code, the special language of computers. But the actual hardware is these gates, which transform zeros and ones. So here we've got an input of 0 and 0, and it's given us an output of 0. This is because this is an example of an AND gate in a computer, and an AND gate's rule is if I have an input from here and here, that is, both numbers have turned to 1, both symbols have gone from 0 off to 1, only if they're both 1, my output will be 1, which is why we've got 1 and 0 here, and this is an output of 0, 0, 1, 0, 0, 0, 0. Here we've got 1, 1, and we've got an output of 1. That's a bit like saying, if neuron 1 is activated here, and neuron 3 is activated here, then yes, we will fire here. So this is an AND gate, basically. So, like a computer, which means that the brain can have rules and codes for understanding the environment. In the case of our cats, with the predator detection module, we can say that if the object is green, it is long, and it slithers, 1, 1, 1, then the output is a snake. There is a snake in the environment. The output is equal to 1. And so there's the logic of recognizing a snake. And that's all good in terms of a computer, but how biologically realistic is that? Well, we need to build a PDM in terms of how it would actually work in the brain. So we're going to start with the retina of the cat, which is found at the back of the eye here. And here we have this optic nerve that sends the signals from the eye towards the brain. So photons of light are bounced off objects in the environment, and they enter the cornea, go through this lens, and are focused into beams onto this back surface, referred to as the retina. Inside the retina, there are individual cells called rods and cones. Rods fire when a photon hits them, and it causes them to send an action potential and a little electrical pulse down their axons. Cones are more specific. They only fire if the photon has a particular frequency, if it's a particular colour, it has a particular energy, which we of course do recognise as colour. These signals stimulate next neurons in the network, which are these bipolar cells. Say when this red cone fires because a red particle of light has entered the retina and hits this red cone, it's saying that there's a point of light in the environment that is red. This stimulates the bipolar cell, which stimulates the next cell in the network, which is the retinal ganglion cell, this guy here. But these bipolar cells are special because they can do something else, which is when they're stimulated by one cell, they inhibit the cells around it. And this just sharpens up the image. It makes a point of light with shadows around it, and this enables you to see features. So we've got dots being summed up, and that's what these retinal ganglion cells send as signals. These signals then pass down nerve fibres from the retina to the lateral geniculate nucleus of the thalamus, featured here, then are passed along another set of nerve fibres to the striate cortex located at the back of the brain. And so on the outside of the brain we have the wrinkly part, the cortex. Now, because vision is processed at the back of the brain, an area called the striate cortex or the occipital lobe, what we can do is we can add put electrodes into the brain. Now this is harmless because the brain itself, because it does all the sensory perception, the brain itself doesn't actually have any sensory receptors in it. Which means if you were to cut through my brain right now, which I'm pretty sure you're probably ready to do with all this scientific explanation, I wouldn't feel it. Because that's the organ I use to feel things with. Which means you can stick electrodes into a brain and it causes absolutely no harm, provided that you don't do any structural damage. And this is what neuroscientists do all the time. And that's what they're doing here. They're putting these black dots are piercing the brain with electrodes to record the electrical activity of individual nerve cells, individual neurons in the striate cortex of the cat. Here you've got the front of the brain, the back of the brain, left and right, and this is the top view.
The researchers actually listened in to individual nerve cells firing in the anesthetized cat as they presented it with different visual images.
When we started working, Torsten and I, in the late 50s, we set up our first experiments, and they didn't go well because at the beginning we couldn't make the cells fire at all. We'd shine lights all over the screen, and nothing seemed to work. And rather by accident, one day, we were shining small spots, either white spots or black spots, onto the screen. And we found that the black dot seemed to be working in a way that at first we couldn't understand until we found that it was the process of slipping the piece of glass into the projector, which swept a line, a very faint, precise, narrow line across the retina, and every time we did that we'd get a response.
So what Huber and Wiesel basically discovered is that these neurons at the back of the brain, they're not responding to points of light like the lateral geniculate nucleus, which is roughly in the centre here under the wrinkly cortex, or even the retinal ganglion cells here. What they were responding to is bars of light, and here we've got an example of a neuron that is very specific. When the bar is orientated this way, you can see here that the neuron is not making any electrical signals. It makes a few as this bar tilts a bit more to the left, and then this increases as the bar orientates to the specific orientation that that neuron responds to. You can see here it's silent, and then it goes, and then that's how it codes that there is a bar of light there at this particular orientation. And it doesn't respond to the other types of orientation. And so this is a bar detector, and this is what we call the receptive field of this neuron. This neuron here specifically responds to this line in this particular orientation. Why does it do that? Well, going back to our idea of neural computation with the McCulloch and Pitts neuron that we discussed earlier, we can show that if individual sensory neurons, in this case neurons from the lateral geniculate nucleus that respond to individual points of light, which of course the lateral geniculate nucleus receives its inputs from the retina, so these retinal ganglion cells here, which represent points of light, their information is preserved in the lateral geniculate nucleus because of its inputs. These represent individual points of light, and they stimulate the next neuron in the network. But this neuron's threshold is so high that an individual input won't activate it. This neuron only activates when several of these neurons fire, and it integrates this, and the sum of their inputs are greater than minus 55 millivolts, which enables this neuron to fire, which means that this neuron will only fire when several points of light are organized in a line in a particular organization. So what we've got here is these X's, which are the retinal cone cells, or rod cells, which together activate those bipolar cells, which activates the sharpening of the image, the on in the center and the off in the surround, as it's called, which sharpens the image. And when you get several points of light with shade around them, we recognize this as a line, like pixels. And that's precisely what this cell can do, because all of these points of light, all these cells that are activated by points of light in a line, are enough to reach the threshold for activating a neuron that represents a line. And guess what? It's turtles all the way up, because this line cell, which we call the simple cell, this line cell is arranged in the next layer of the brain, which is called the visual area 2, or extrastriate cortex. These line cells can then activate another complex cell in the next layer of the network. And so this might recognize curves or shapes. And this is precisely what we find in cats. And this is a representation of what we know about cat vision. So we've got the retinal ganglion cells, which send their signals to the lateral geniculate nucleus. The rod cells, which respond to any light, will take this, what we call the magnocellular pathway. This has no representation of colour, but it's very good at recognising movement. And this sends this signal to the primary visual cortex, the striate cortex, which have the simple cells. These guys are located in this next layer of the brain. Simultaneously, the coloured cells, or the cone cells, take the parvocellular pathway, these other layers in the lateral geniculate nucleus, which contain colour information, points of coloured light. These cells here send their axons here to the next cell. And so these parvocellular layers represent bars of light, but with specific colours. And they're arranged in wheels which we call retinotopic pinwheels. And then these go up to further layers, up to layers such as the visual area 4 and the medial temporal lobe, which I can show you. The medial temporal lobe is located around the sides of the brain, here. And these are so high up in the hierarchy that they can recognize objects, which, if the light hitting the retina is, for example, green and in a line and perhaps a bit wrinkly and smells funny, then up here, the medial temporal lobe and V4 will categorize it. The neurons will represent a cucumber. We can see this phenomenon happening in humans as well with memory. So in our medial temporal lobe, we can store experiences as memories in much the same way. And here we're recording a neuron from a man who's got epilepsy. So he's having surgery on his brain, and he's awake. But as I said, you don't feel pain in your brain. So he's got no problem with being awake. What they're doing is they're recording his neural activity from one neuron whilst showing him images. And so we can see here the neuron doesn't respond to a video clip of the Titanic. It doesn't respond to US stock exchange values, but it responds to The Simpsons specifically. It doesn't to the I Have a Dream speech by Martin Luther King. It doesn't respond to the Pyramids of Giza, but it gets very excited electrically by The Simpsons again. So this neuron is obviously a part of a network in a layer that recognizes The Simpsons, which is, I think that's really amazing that we've got a Simpsons neuron. There's been reports of neurons of particular celebrities like Jennifer Aniston. There is a Jennifer Aniston neuron in the human brain. Isn't that amazing? But why do cucumbers activate a neural pathway which is intended to recognize snakes? Why is the predator detection module activated by snake-looking inputs? Well, the evidence is actually provided by behaviour of the common toad, where prey catching is elicited by innate prey detection neural selective mechanisms like our predator detection module for snakes in cats. As you would expect, this guy has detected a worm in his visual field. He approaches it, fixates, and eventually the prey is consumed when a neural signal activates the muscles of the tongue and jaw. He's numbing on that quite nicely. And this occurs for all stimuli that are worm-shaped and have a worm-like movement. Okay, so to use an analogy of how a cucumber might activate the snake recognition module in cats, we could use this super mega feeding platform thing to mimic the sensory features of perceiving a worm by producing an object that is in worm configuration and that elicits the innate releasing mechanism of the toad, which is what you can see here with this horizontal black line. We can also flip it round so that it looks like, so it becomes a vertical line. And as you can see here, the horizontal line that moves in a way that mimics a worm elicits the innate response, whilst a vertical stimulus goes completely unnoticed by the toad. Thus, we can hypothesize that any object that excites a neural network in the same way that the subject that the neural network has evolved to process will elicit a particular innate behavior, like a cucumber exciting a snake network. And we can test this with recordings.
The toad's brain consists of the telencephalon, diencephalon, mesencephalon, cerebellum, and myelencephalon. Retina and optic nerve represent, embryonically, a protuberance of the diencephalon. In the retina, an area of receptor cells, via interneurons, is connected to a ganglion cell. The region of the visual field it corresponds to is called receptive field. This is the receptive field of a ganglion cell whose axon, travelling in the optic nerve, contacts neurons in the optic tectum of the contralateral midbrain or neurons of the pretectal thalamus of the contralateral diencephalon respectively.
So as you can see, the optic tectum and the pretectal thalamus are just like the lateral geniculate nucleus of the thalamus in the cat's brain.
When an object is moved through the receptive field of such a neuron, its impulse response can be recorded by means of a microelectrode made audible and visualized on an oscilloscope. By means of a perimetric apparatus, visual stimulus patterns corresponding to the behavioral experiments can be moved on a belt in different directions through a neuron's receptive field.
I always think this bit looks so bizarre. It looks like some sort of space docking thing for like a lunar module or something. Like, this toad is probably just like, what am I looking at, man?
During the recordings, the toad is anesthetized and pharmacologically immobilized. First recordings from a neuron of the optic tectum. To a worm-like moving stripe, the neuron fires with high impulse frequency. The same stripe in anti-worm configuration remains practically unresponded.
And so basically what I'm proposing is that if you were to record the cat's cortex whilst it's looking at a cucumber, and if you knew the exact neurons that were forming the network that I'm suggesting is the innate predator mechanism, recognition mechanism, or detection mechanism, then you would find that the cucumber is activating that innate predator detection module. So how do these neural circuits or psychological mechanisms like the predator detection module in cats, which responds to the cucumber, how do they actually evolve? How does a mutation lead to a psychological ability or cells to be connected in such ways that they can represent snakes, and that they do it automatically and with blindingly fast computation to get the cat the hell out of there? Well, to understand that, we've got to understand the molecular biology of how neurons are formed. Neurons are formed from stem cells early in development, at approximately 6 weeks onwards. But these neurons, when they form from stem cells, don't have the dendrites, and at that point they don't actually have an axon either. What they do is when they start to form axons, they have this end called the growth cone, and it has these fingers called filopodia that stick out of the end of the axon in the growth cone. And what the filopodia do is they sense the environment around itself in the early brain tissue for chemicals that tell it where it needs to go. Because if you think about it, in the giraffe brain, sensory signals from its feet have to go all the way up the axon, all the way to the central nervous system in its brain for it to understand where its feet is. In the giraffe, that is several meters. That is several meters for this axon to travel. So the axons need to have a very particular guiding system. An example of a chemical that acts as these guiders is netrin 1. Netrin comes from the Sanskrit word netir, which means one who guides. And here we have a small bit of membrane. This is the sac of the cell, if you will, with the chicken soup inside that I mentioned earlier, all the salty insides. This membrane is the outer surface of the cell, and on the filopodia, that's going to be surrounded by membrane. And embedded on the outside of this plasma membrane is receptors. This is the DCC receptor, which stands for deleted in colorectal cancer receptor, and that's just named because we discovered this receptor and during its involvement during a colon cancer event. What netrin does is it's secreted by other cells in the extracellular makeup around the neuron, and what netrin does is it guides the axon by binding to DCC, which sends a signal down into the cell which changes the cytoskeleton of the cell. What is the cytoskeleton? That's this thing here. That's the interior that extends the cytoplasm, the cellular membrane out from this growing growth cone and out into the environment around it in these long fingers called filopodia. So when netrin binds to this receptor, it encourages these fingers to extend outwards and to grow the growth cone. Here we've got the DCC receptor with an UNC5 receptor. When these go together, what they actually do when they bind to netrin is they do the exact opposite. They break down these bundles of actin, of the cytoskeleton. The cytoskeleton is composed of these little actin subunits that bind together. This is a very complex reaction. I'm really under-exaggerating all the processes that are going off here. But so when it binds to netrin, when it's got these receptors, what it's doing is it's preventing these from extending, and that's what controls the growth of the cone and this neuron, for example, from establishing a connection with this neuron, the dendrite of this next neuron. And so by having particular, very, very specific receptors and very specific signaling molecules, you can build very precise networks. Now, of course, both the receptors, the combination of receptors, where they go, and the signaling molecules are controlled by your genes. So if you have a genetic mutation in any of these components, you have a genetic mutation in the network formation, which changes the computation, which is how the brain, through mutation of its genes, can give rise to these various computational abilities, the tools on the Swiss Army knife, which we pass on to the next generation. And of course, that is how our predator detection module inside the cat is passed on from generation to generation, and why it gets activated by looking at cucumbers. Now, is there evidence for this in humans? Yes, there is. Not for predator detection, but something far more interesting. Back in the 1950s, or even before that, the early 1900s, the field of psychology was dominated by a thing called behaviorism. This is your Pavlovian dog conditioning thing, which is basically if you pair a one stimulus with another repeatedly, you learn an association between the stimulus. In Pavlov's dog's condition, this was a bell with the presentation of food, so that the dog learns to expect food when it hears the bell. Or another form of conditioning of behaviorism was operant conditioning, and this is when you get given a reward for a behavior. And the idea of a behaviorist, and if you just take that all behavior is socially learned, then you should be attracted to what rewards you. And so the idea was that in humans, the reason why we develop a relationship with our parents is because they feed us. So big behaviourists like B.F. Skinner would think that this is the ideal mother. If you totally believe that everything in your environment is socially learned, then this is what you think your mother should be. It should just be a bottle of food. And this is Harry Harlow's famous experiment in the 1970s, where he made a wire mesh mother for rhesus macaques. And in a different compartment, he built a model which looks more like a rhesus macaque and has a nice fluffy pelt but provides no food. So this one's more like the mother stimulus. This one's less like the mother stimulus, but provides food. So if you're a behaviourist that believes that everything is socially learned, this is what you think your ideal mother is. However, if you have an evolutionary psychologically adapted mechanism for recognizing mothers, because mothers increase your chance of survival, which increases your chance of reproductive success and passing on genes to the next generation. This is the mother you should be innately predisposed to liking, because it makes you feel happy. It's more like your mother. And this is what he found in rhesus macaques. His rhesus macaques preferred the fluffy mother stimulus model far much more than they preferred the wire mesh feeding bottle. And this just goes to support it. And in the end, he actually had to put the wire mesh mother in the same cage as the felt mother, because the infant was actually preferring to have comfort from this mother stimulus model than actually feed itself. And you can even see here it's actually stretching across from its preferred model just to try and get food, and then it would instantly climb back to its mother. And in human babies, we can see that this happens super early. Even 1 to 2 days after birth, infants prefer to look at images that resemble the human face, and we've actually discovered the brain area that carries out this computation, and it's in the inferior temporal cortex. So that is a summary of evolutionary psychology and, of course, the death by cucumber phenomenon. Thank you for watching.